Welcome to the Carlin Sales Pro Green Plus BuyerFest 2020. Uh, the topic of this presentation is called Biological Control, What You Need to Know. My name is Raymond Cloyd. I'm a professor and extension specialist in horticultural entomology at Kansas State University in the Department of Entomology. So I want to give you an overview of what to expect during this presentation. I'll give you a brief introduction. We'll cover the basics of biological control. We'll cover the biological control agents. They're also referred to as natural enemies. And then I'm going to cover how to implement a successful biological control program. So first of all, greenhouse-grown horticultural crops are susceptible to a wide range of insect and mite pests, and again, many times simultaneously. That is why we talk about multiple pest complexes as opposed to individual pests. And that may be an issue when you're using biological control. Most of greenhouse production system involves polycultures or multi-crops grown simultaneously. Consequently, this results in a multitude of different insect or mite pests that may be encountered. Well, the primary means of dealing with insect or mite pests in greenhouse horticultural cropping systems is still the use of pesticides, and I'm going to focus on insecticides and miticides. Well, why is that? Well, they're cost effective in general, easy to apply, and there's that psychological satisfaction that after you made an application, you have killed or nuked a lot of bugs, in theory. Well, what about an alternative plant protection or pest management strategy? And that is biological control. Biological control has been around since the 1970s and is another practice or strategy to deal with insect or mite pests in greenhouse production systems. Well, here is an example of containers that you can buy from suppliers or distributors that contain what we call the biological control agents. Let's talk about what biological control is. The use of biological control or natural enemies, such as parasitoids and predators, are designed to regulate insect or mite pest populations. Biological control agents will not eradicate an insect or mite pest population. The success of biological control is contingent on maintaining insect and or mite pest numbers at levels low enough to minimize plant damage. Biological control is a preventative strategy. Once the pests are there in outbreak proportions, it's too late for biological. In most cases, you should release your biological control agents before you actually see the pests. The key is regulation of insect and mite pest populations. Here's an example we call mass release. These are predatory mites on a formulated popsicle stick that are placed in individual trays of these plants. This is what we call inundative use. You're using the biological control agents like a biological insecticide. So what are some of the requirements of a biocontrol program? You need to know availability of the biological control agents or natural enemies. You need to understand the biology, the life cycle, and behavior of insect or mite pests within cropping systems. You need to scout crops regularly and order your biocontrol agents as needed. We generally recommend ordering them 2 weeks in advance prior to release. And then you need to follow up to assess success or failure. So there are a wide variety of biological control agents commercially available, and these are just the way they're packaged upon receipt, or the way you would get them from the biological control distributor or supplier. Let's first talk about the types of biological control agents. The three are parasitoids, predators, and beneficial nematodes. So on the upper left is the parasitoid, the predatory mite, and then the beneficial nematodes. These are the primary biological control agents that you can purchase from suppliers and distributors. Let's talk about the categories of biological control agents. The first are specialists. Those feed on only one insect or mite, prey, or host, or particular life stage. There are some that only attack the egg, larva or nymph, or the adults. Most of the specialists are parasitoids. The second category is generalist. They feed on a variety of insect or mite, prey, or host, and also tend to feed on different life stages. They'll feed on the egg, larva, nymph, and or adult. Most of the generalists are predators. Not all, but most of them are. Okay, so let's talk about parasitoids. Ephidus aerivii, Ephidus colmani, these are both aphid parasitoids. Incarcia formosa is a parasitoid of the greenhouse whitefly. On the bottom right, you can see this is a whitefly pupa that's not been parasitized, and the one next to it in black, that indicates that has been parasitized by Incarcia formosa. Well, how does this work? Well, a female parasitoid inserts her ovipositor or egg-laying device into an unsuspecting host, in this case, an aphid. The egg is laid, the egg hatches into a larva that starts eating the inside of the aphid. Well, eventually the larva pupates and then becomes an adult. Then the adult, with their chewing mouth parts, would create a hole at the back of the insect and emerge out. The time it takes to go from egg to adult depends on temperature, but it can be 7 to 14 days. Now, what you want to see is this. These are parasitized or mummified aphids. This demonstrates the success of the program. You can see that some of these aphids have holes in them. That's where the adults have come out. Now, if you're still having problem understanding this concept, I'm going to go to Hollywood and show if you've seen these two movies, then you know what a parasitoid is. If you've seen either Aliens or Aliens 2 or Aliens, that's an example of a parasitoid. Well, how do you release them? Well, you can get containers which contain the parasitized or mummified aphids, place them in the crop area near aphids, and let them do the job. Or you can get these what we call release cards, and on them are the pupa, in this case whitefly glued, and you can see the black parasitized whitefly pupa. Very shortly, those will have adults emerge, and the adults will start looking for whiteflies, the females in this case, to lay eggs. So what do you do with these? Well, you hang them on branches, and again, here's where the parasitized pupa are. When the adults come out, they'll start searching for whiteflies and for the females to lay eggs. Now, what's really important when you release parasitoids, you got to take down your yellow sticky cards because the parasitoids are attracted to this wavelength of yellow and they will be captured. And basically what that does, it defeats the purpose of your biocontrol program, in this case, releasing parasitoids. You want to leave them down for about 7 days, and then you can use them again for your scouting purposes. Well, let's talk a little bit about aphid parasitoids. The Ephidus aerivii attacks what we call large aphid species, in this case potato and foxglove aphid. Ephidus colmani parasitizes small aphids, in this case green peach and cotton melon aphid. Aphelinus abdomalis parasitizes potato and foxglove aphid and some other aphid species. Aphidius matricaria parasitizes green peach aphid. So what's really important to understand is you have to have your aphids identified before you select a specific parasitoid. Because if you have green peach, you don't want to release Aphidius aerivii. You want to release Aphidius colmani. Well, What if you have more than one type of aphid? Well, then you can get a mixture of different parasitoids, and this is a mixture of Aphidus colmani and Aervi. Aphidus colmani will attack the green peach and melon aphid, whereas Aphidus aervi will attack the potato and foxglove aphid. Now let's switch to talking about predatory mites, another major group of biological control agents used in greenhouse production systems. We have Neoscelis californicus, Neoscelis cucumaris for western flower thrips, and of course Phytoceus persimilis, which has been around since the 1970s for regulation of two spotted spider mite populations. Well, there are a lot of commercially available predatory mites. Some will attack more than one mite or insect. Some are very specific. Phytoceus persimilis will only feed on two-spotted spider mite, where some of the others on this list, like Andersoni, Amblysseus andersoni, will feed on two-spotted spider mite, tomato russet mite, and broad mite. Well, here are the way they're formulated. You can get them in these containers, or what I like to recommend are these sachets where you can hang them on the plants or insert them in the growing medium, like we call these popsicle sticks, put them right near the foliage, and the mites will come out, get on the plant, and start looking for prey to feed upon. Here is another container. This contains Stratolalius semitus, formerly known as Hypoacis miles. This is a predatory mite primarily for fungus gnat larvae that are in the growing medium. Well, this is the way you apply them. It's called the salt and pepper shaker method. And you can see this is a vermiculite carrier, and that's an indication where you made your application. This is very, very labor intensive. Here's an example of a container of Phytocelis persimilis placed along flats. Look how small those plants are. This again emphasizes that biological control is a preventative strategy. Well, again, this is labor intensive. However, we have new we'll talk about that, mechanized means of releasing these predatory mites. But the sachets are very efficient. You put them in the growing medium, the mites come out in about 2 weeks, and they'll start looking for food on the plants. These last about 6 weeks before you can remove them. Now, you want to keep them away from direct sunlight, and more importantly, don't get them wet, because that will result in mold, which could negatively affect the predatory mites. Well, I mentioned that it's very labor intensive to do the salt and paper shaker method, but now we have mechanized means using blowers that are modified or these new devices. What this is you put the bran with the mites, and through the Venturi effect, it blows them across the greenhouse. This is much more efficient and labor saving than the old salt and pepper shaker method. What happens is the mites are traveled across, they land on the plants, and then start looking for food. The research that has been done at Kansas State shows that the mites do survive this method of application. Okay, in addition to predatory mites, we have two other biological control agents. We have the insidious flower bug, and we have the row beetle. The insidious flower bug is a predator of western flower thrips, and there it is, the size compared to a dime, and there's a close-up of the predatory Oris insidiosus adult feeding on a soft-bodied insect. Looks like this case it's whiteflies. Now, the row beetle adult on top and larva are both predaceous. They reside in the growing medium, and they feed on fungus gnat larvae, and based in our research at Kansas State, they will feed on the prepupa and pupa of western flower thrips, which are located in the growing medium. The third group of biological control agents are the entomopathogenic or beneficial nematodes. Nematodes are microscopic roundworms. They need moisture to move around. So when you make an application to the growing medium for control of fungus gnat larvae, the nematodes enter the larva through natural openings, the spiracles or breathing pores, the mouth or the anus. They then release a bacteria, and it's actually the bacteria that kills the insect by consuming the internal contents. The nematodes use the residue as a food source. And then it goes through a series of generations of infective juveniles afterward will break out of the dead insect and start looking for another host to attack. So this is an example of a dead insect, and these are billions of nematodes out there. If they find a host, they will infect it. If they don't, they may die. Remember, nematodes need moist conditions for survival. Here is a product called Nemesis that is registered for fungus gnats and also western flower thrifts. This is very important. You're supposed to get 50 million nematodes. Be sure you count them before you release them to ensure that you have the appropriate number. Just kidding. You really don't have time to count 50 million nematodes, but we will talk about quality later on in this presentation. So how do you succeed using biological control agents? We've talked about the different types, parasitoids, predators, and entomopathogenic nematodes. Now, how do you use these and succeed in your biocontrol program? Well, you need to establish a reliable scouting program. You need to correctly identify the primary insect or mite pest. Remember when I talked about aphid parasitoids? You need to get the aphid identified before you actually know what parasitoid to purchase and then release. Identify available biological control agents for specific insect or mite pest. Establish a relationship with a supplier of biological control agents. Minimize pesticide residues. Before you start a biocontrol program, you cannot spray any pesticides, because some of the broad spectrum pesticides have residues that may negatively impact the biological control agents. You want to start clean, you want to implement good sanitation programs, and you want to order your biological control agents early. I recommend about 2 weeks prior to release. And then you want to assess the quality of the biological control agents. You want to release them immediately upon arrival. You want your biological control agents to be released in the morning or early evening. You want to introduce your biological control agents before insect or mite pest populations are high or reach outbreak proportions. If you have already established aphids, whiteflies, or thrifts, it's too late to release biological control agents. And then evaluate performance of the biological control agents at the end of the growing season. Well, let's talk about scouting. Before you even consider implementing a biological control program, you have to have an established, reliable scouting program. Scouting involves using yellow sticky cards and doing visual inspections, because spider mites, eggs, larvae, pupa will not be captured on yellow sticky cards, as well as mealybugs. They will also not be captured. So you have to do a combination of sticky cards and visual inspection. Well, here's an example of a grower I deal with. They are identifying the insects using a 16X henlands, and then they're recording the data. Well, what are they recording? Well, here is the months of March and April, the date. They're recording the number of thrips, whiteflies, mites, foxglove aphids, and aphids. What this is a snapshot, but if you do this regularly, you can assess the population dynamics or the numbers of insect and mite pests during the course of the growing season. And if you start seeing increases, then you need to release your biocontrol agents early. If the numbers get out of hand, then you may have to use an insecticide or miticide. Well, let me talk about these are the three factors that can influence the success of a biological control program: quality control, environment, and plant characteristics. For the sake of brevity, I want to focus in on quality control. The quality control issues associated with biological control agents. When you order your biocontrol agents and then you get them, you do not want to assume that they're alive. The data is out there unequivocally that dead insects will not regulate populations of live insects. So the first thing you want to do is make sure that they're packaged properly, either in styrofoam with newspaper and peanuts to make sure they're not jostled. So the first thing is make sure that the packaging looks okay. So here's a shipment, but now you have to ask yourself the question, are they alive? Okay. Not only do they have to be alive, but they have to be functional. That is, they have to go out there and find the pest. So these are just some examples of products that you can obtain from the suppliers of natural enemies or biological control agents. Here's an example. Are all the predatory mites alive, and is the number similar for each sachet? Have you ever asked yourself that question? Well, what you can do is quality control. Here we are taking some of the popsicle sticks, which I mentioned earlier, and we break them open. And we start looking for predatory mites. Now, remember, there is a grain mite that will serve as an alternate food source, so don't confuse that with the predatory mite. Basically, what we're looking for is some level of activity. Are mites moving around? If not, then they may be dead and you need to contact immediately the supplier or distributor. Here is a really easy way to determine if the sachets contain functional mites. You bend the stick a little bit, you place it on a yellow sticky card, and the mites will either fall onto the sticky card or they'll travel down and get captured. This is a really easy, quote, quick and dirty way to assess the quality of the sachets. Well, what about parasitoids? Well, what we do in our lab, we take mason jars, we put a section of a yellow sticky card, about a 1" by 1", attach it to the lid, and then we place the release card, one of them, in there. We cover the mason jar and we wait about 48 hours. If the parasitoids are alive, they will be captured on the yellow sticky card. That tells you that that shipment probably has got alive and functional individuals. Well, what about nematodes? Well, what you do is take a sampling of the product, you put it in a container of water. You can take a jar, fill it half full of water, place the material in there, shake it up gently, and hold it up to the light. If you see nematodes that are curling and moving, they're alive. If they're straight out, they're dead. And again, dead nematodes will not kill fungus gnat larvae. When you do encounter difficulties, either dead insects or you don't get the number you ordered, you need to contact your supplier or distributor immediately. Well, what about storage? I don't recommend storing for very long, no more than 24 hours, but this is an operation we deal with that uses a lot of biological control, and you can see the volume and different types of beneficial insects or natural enemies or biological control agents. They store them in a refrigerator and no more than 24 hours and then release them. Well, What life stages, egg, larva, nymph, pupa, or adult, are susceptible to natural enemies? Not all the predatory insects or mites or parasitoid are going to attack all the life stages. Well, let's look at western flower thrips. We have Neocoelus cucumaris, which attacks the first end star, Amblyssia swirsky, which attacks the first and maybe the second. There is no biocontrol agent commercially available that attacks the eggs because they're inserted into the plant tissue. Then we have Orris insidiosus, the insidious flower bug. It attacks both the adults and nymphs, first, second instar, and also the adult. Stratiolaelips simitus, also referred to as Hypoapsis miles previously, attacks the pupal stages. This is the prepupa and the pupa stage of the western flower thrips. These are in the growing media. The adult first and second instar feed on the foliage and flowers, that is the above ground plant parts. And then Delocia, also referred to as the Theta coriara, the roe beetle, attacks the pupa. And in our studies at Kansas State, we have found that the adults will feed on the prepupa and the pupal stage of western flower thrips. So, can you succeed using biological control in greenhouse production systems? Yes, you can, but it takes a different mindset compared to just going to the container, grabbing an insecticide, and then spraying. Okay, but it can be done. Well, what about some information? Working with some colleagues of mine in Michigan State University, we developed a fact sheet called Commercially Available Biological Control Agents for Common Greenhouse Insect Pests. And what each page has is the commercially available biological control agents for the specific pests. This is aphids, has all the parasitoids, some of them we've mentioned, and the predators. We then provide guidelines about the optimal temperatures, humidity, release rates, and things that help you succeed when implementing a biological control program. You can download this online and get a PDF. Well, in addition, we have a biological control online course called Biological Control Course for Greenhouse Growers. Working with Heidi Lindberg in Michigan State University Extension, we worked, and this is now the third year of the course. It contains 5 units. Each unit is independent of each one, and you can register by getting on this website. There are 2 registration periods, fall and summer. It's a minimal cost of a little over $120. Here are some suppliers of biological control agents. These are ones I've worked with for many years. I feel very comfortable. Now, the last one is a new one I have not worked with called BioBee, but they do have a wide diversity of biological control agents available. So if you want to try a new one, you might want to look at BioBee. Well, that is the end of this presentation. I want to thank you for your participation. And if you have any questions or comments related to this presentation, that would be greatly appreciated. So with that, I am done.